Hey everyone, today we're going to continue on with the nervous system. And today is going to be a kind of mostly lecture kind of day, so there's not going to be any Schoology assignment. And we're going to focus a lot on nervous tissue and the structure. So to begin, nervous tissue is made up of two types of cells. The first one is supporting cells, and the second one is neurons. So supporting cells actually resemble neurons. However, they are unable to conduct nerve impulses, which means they do not send out signals. And another thing about supporting cells is they never lose their ability to divide. So they're always undergoing mitosis to create new cells. Neurons, on the other hand, these are often called nerve cells. These are going to be the ones that conduct the nerve impulses. They send signals from our brain to our muscles. And these actually, if you damage them, can lose the ability to divide, which is why they tell you don't ruin your brain or don't have injuries to your spine, because if you damage them enough, they're not able to basically regenerate. So we're going to first talk about support cells, and we're going to focus on support cells in our CNS. So remember, our CNS is our central nervous system. These are often grouped together and they're called neuroglia, and they're also called glial cells. So either one of those you hear, these are support cells within your central nervous system. The general function is to support, insulate, and protect the neurons because, again, if neurons get damaged, this can cause a lot of issues within the body. So let's talk about the different types of supporting cells in your central nervous system. The first one we're going to talk about are called astrocytes. These are very abundant and they're star-shaped, hence the name astro, like astronomy. What they do is they help brace and anchor neurons to the capillaries, which are just blood vessels. So these are just tiny blood vessels. They help provide a barrier between your blood vessels and the neurons. And the function of this is to protect them from any harmful substances that may be in your blood, because you don't want those harmful substances to get into things like your brain or your spinal cord. They also help control the chemical environment of the brain as well. Here's a good example of an astrocyte where it's connecting to a blood vessel. So here's your capillary, and then it connects over here to your neuron as well. The second type we're going to talk about is microglia. Microglia are said to have a spider-like appearance, and they are phagocytes. Phago, the key word here, is to eat. They help monitor the health of neurons by basically ingesting or eating any dead debris, such as dead brain cells and bacteria. Here's a good example of an artist drawing of microglia cells. These, again, have more spider-like appearance, and they're going to be cleaning up any debris that they find. The third type is epidermal cells. These cells actually line the brain and spinal cord cavities, and they have what's on them cilia, which are projections. So these are tiny projections on the cell surface. And they help circulate something called cerebral spinal fluid. Now, cerebral spinal fluid is a fluid that provides a watery cushion around your brain and your spinal cord. So here is our epidemial cells. Up here, these things right here, this is going to be your cilia that help move the fluid within the brain and spinal cord cavity. And the last type of central nervous system supporting cell we're going to talk about is called oligodendrocytes. These are actually one of the most important ones we talk about, so I'm going to go ahead and star that. And what these do is they wrap around nerve fibers within your central nervous system and give it a little bit more protection, and also help produce something called myelin sheaths. Myelin sheaths we're going to bring up, but basically these help protect but also help speed up the electrical impulses that are sent down a neuron. So here is an oligodendrocytes that's covering a nerve fiber, and they just provide that myelin sheath or that covering. There are a few supporting cells within your peripheral nervous system or your PNS. So again, remember, PNS is your peripheral. The two that we're going to talk about is Schwann cells, and I'm going to go ahead and star this one. These form myelin sheaths as well, but again, these is in the PNS. And then you have satellite cells. Again, these just help protect and cushion. Here's an example of your satellite cells and your Schwann cells. So your satellite goes around the cell body of a neuron, and Schwann cells form the myelin sheath that cover the nerve fiber. Now that we've kind of talked about supporting cells, let's delve into how a neuron is structured. Well, just to clarify, neurons are commonly called nerve cells, and they function to transmit messages, or in other words, nerve impulses, from one part of the body to the other. So how does us seeing a step in front of us transmit to our brain, and how does that get transmitted to our foot to step up? Well, there are a lot of major regions within the neuron, and we're going to talk about several. The first two distinctions is the cell body versus processes. The cell body is the big middle region where your nucleus is going to be located, and this is the metabolic center of the cell. The next major thing we're going to talk about is the processes, or basically the fibers that come off of the cell body. So let's first talk about this cell body. The cell body is, again, the metabolic center of the neuron, and it has a very large nucleolus in the middle. Remember, your nucleolus is responsible for making ribosomes. These contain all the normal organelles that are found in cells, except for they lack centrioles, which goes to further explain the amitotic nature. Amitotic means it does not divide. There are also specialized rough ER, and these are called Niesel bodies, and they also have specialized intermediate filaments that help maintain the cell shape of neurons called neurofibrils. Here is a common structure of a motor neuron. I'm going to hand this picture out in class. You guys are going to study it, and there will be a quiz coming up in a few class periods. But I'm going to go ahead and we're going to define each of these things. First and foremost, the one thing we should know is that the middle of this structure is called the cell body. So that's an important term. In the very middle, there's your nucleus. So I'm going to go ahead and circle that. From there, you have several processes that come off of it. The first process are the ones that stick out over here, and these are going to be called dendrites, and we're going to go ahead and define that here coming up in another slide. The dendrites will eventually go into something called an axon hillock. We're going to define that. It's going to go down this yellow axon, we're going to define that, which are covered by Schwann cells, which form myelin sheath. But there are gaps between the Schwann cells, and we're going to define nodes of Ranvier, and we're also going to define axon terminal. So I'm going to define these, and we're going to come back and look at this picture. So here's just a close-up picture of ones we've already talked about. Again, the big middle region is our cell body, and then the nucleus is in the middle. So let's first talk about those processes, fibers that extend off of the cell body. I said there are going to be two. The first one we're going to talk about is known as dendrites. These conduct impulses towards the cell body, and neurons can have hundreds of dendrites. So up here, if I was going to look at this picture, these arrows are pointing in to signify the message going to the cell body. So all of these extensions here, 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 these are all dendrites that are sending signals to the middle of the cell body. Axons, these conduct impulses away from the cell body, And oftentimes, neurons only have one axon, and it stems from something called an axon hillock. So if I go up here, your axon hillock is right over here. And the axon, there's only one of them. Here's your axon hillock. It goes down and extends. So you have one thing coming off of it. This yellow strand is your axon. Now, oftentimes axons end in something known as axon terminals that we mentioned in the muscular system. And these axon terminals contain little vesicles that have neurotransmitters. Specifically, in the muscular system, we learned how that neurotransmitter was called acetylcholine. Now, axon terminals are separated from one neuron to the next from something called a synaptic cleft, or sometimes called a synapse. We learned about this again in the muscular system. This is the gap between the axon terminal and the next neuron, because neurons, just like muscles and neurons, never actually touch one another. So again, if I'm going back up to this picture, if I go down, here's my axon terminals, the very end. If there was another neuron here, you would see that they would never physically touch, and there would be a little gap here called the synaptic cleft. So again, neurons have hundreds of dendrites, but only one axon. With that being said, I said that we would talk about something called myelin sheaths. So myelin is a short name standing for myelin sheaths. All this is a white fatty material that covers the axons, and they protect and insulate the fibers. The other thing it does is it helps speed up nerve impulses that are being sent down an axon. So in the PNS, the support cells that make up the myelin sheath are known as Schwann cells. And these actually take several Schwann cells to create a myelin sheath. So they have little gaps in between them called nodes of Ranvier. So the picture that I showed you up here, these are Schwann cells. They wrap around the neuron, but there's little gaps. And again, these little gaps, jumps here, are your nodes of Ranvier. In the CNS, these are made of oligodendrocytes, and these are a lot longer, so one oligodendrocyte can form and spread across a large area and can form several different myelin sheaths. So here is an example of a Schwann cell wrapping around an axon and providing that cushion. And basically, that's the end of the lesson. I know if you were confused on anything, this is some new terminology. Please let me know. Please feel free to reach out, and I'll help you to the best of my ability. And if you have any questions, just please make sure you email me. Otherwise, I hope you guys have a great day.